Hi, I'm Mike Hutchins, Extension Dairy Specialist at the University of Illinois. This module will discuss the basics of the digestive physiology of ruminants. Let's first look at the different compartments of the ruminant stomach. Remember, there's only one stomach with 4 compartments. The first major structure is the rumen. In the adult dairy cow, about 57% of the volume is found in this portion, also called the fermentation vat, the common name. In a calf, however, it only makes up about 30% of the volume. You'll see why this changes a bit later. This, of course, is the major structure also that will bloat if there's abnormal fermentation going on in the rumen. The second compartment would be the reticulum. It makes up about 7% of the cow's total volume, also called the honeycomb area, and makes up about 8% in the calf. Another common name for this one would be called the hardware area, or the hardware trap, because this is where wire and pieces of metal would be caught, and also where a magnet would be found if given to a cow to retain long, sharp material. The third compartment is the omasum. It makes up about 24% of the cow's volume. The common name called the many plies, looks a little bit like a phone book to be exact, and makes up about 13% of the volume for a baby calf at birth. Not many things can go wrong with the omasum except for compaction, due to such things as very fibrous materials such as sunflower hulls or large amounts of mineral being consumed. The fourth compartment is called the abomasum. It makes up about 11% of the cow's total rumen structure and is also called the true stomach. Here's where the first secretory tissue would be found. You'll find some enzymes to digest protein. In the baby calf, it makes up about 49%, or almost half, of the whole volume. Thus, the baby calf is a bit like a monogastric, unlike the ruminant itself. Probably the most common disorder you and I will hear about with the abomasum would be the displaced abomasum, or twisted stomach, which means this stomach leaves the bottom right side of the cow and pops up 90% of the time on the left side towards the dorsal or the top part of the cow. Now let's go back to the baby calf. How does this calf's small, immature rumen develop? Basically, it is caused by stimulation of the rumen papillae, primarily by feeding it grain. This grain will produce propionic and butyric acid, and this causes stimulation of these small finger-like projections called papillae. These will absorb VFAs and are stimulated by the initial VFA production in the baby calf. How do these rumen microbes get into the calf? Well, generally they're found in the environment. Usually they're from older heifers and cows. So if the baby calf is exposed to the environment of other animals, she will generally pick up these rumen microbes and establish in the rumen. We will then feed forages to this young calf to develop the musculature of the rumen. Baled hay, long forages cause this organ to develop a much stronger muscle contraction to allow her to digest and move feed around. It normally takes about 4 to 6 weeks from the time the calf is born before the rumen is really functional, which means you'll see the baby calf chewing its cud, eating forages, and start seeing the rumen development and formation of the papillae. In an adult animal, we have the process called rumination. Rumination is cud chewing, and 4 basic processes will occur. When there is a tickling around the opening of the rumen, this will cause the animal to regurgitate or pull up a bolus of feed. This bolus of feed then comes back to the animal in its mouth, and she will re-chew this. As you'll see in the second bullet item, she will chew at least 30 to 40 times per cud if it is a strong cud and has long particle size. This is very important for proper rumination to occur. While she is re-chewing, the third R occurs, resalivation, which means we mix saliva, which contains buffer, in with the feed, which then can be used to buffer the rumen. Once the cow has chewed this fine enough, much like you and I chewing our own feed, she will then re-swallow this bolus of feed back down into the rumen, and the process will start over again. She will spend nearly 500 to 600 minutes a day chewing time. This is important to have proper rumen fermentation and adequate amounts of saliva being produced in the dairy cow. Once the feed is fine enough and is heavy enough, it will then leave the rumen. That determines when it can move out into the reticulum and other parts of the digestive tract. A second process in the rumen is called eructation, which is a nice word for saying belching. Eructation is the removal of excess fermentation gases from the rumen. The 2 most common gases will be methane, that's blamed for the greenhouse effect from time to time, and carbon dioxide. If this cow cannot eructate this gas up, she will then bloat, and of course requires quick medical attention. Rumen characteristics. If you and I took a sample from the rumen, we'd expect the pH to vary from 5.9 to about 6.5. More about factors that cause this shift to occur a bit later. Every 20 to 40 seconds, if we had a stethoscope, we'd expect to hear a rumen contraction. This is normal because it mixes the feed, causes the cow to chew her cud, and also relieves gas. Finally, the rate of passage of feed varies from 6 to 9%, depending on its particle size and the type of feed it is. For example, forages are quite slow. Straw would be very slow, where corn grain would be very, very fast, especially if we ground it very fine. If the rate is too fast, we can have abnormal fermentation. If it is too slow, it reduces dry matter intake. We then look at the actual fermentation going on the rumen. We'll have three primary acids. These are called volatile fatty acids, or VFAs. It's obvious if you open up a cow and smell. It has a very pungent and disagreeable odor coming out of the rumen. The major one will be acetic acid or acetate. Either term would be correct. This is a 2-carbon volatile fatty acid and is the major one found in the rumen, making up typically 60 to 75% of total VFAs, and is a major source for energy for the dairy cow and a source of milk fat. The second VFA is propionic acid or propionate. This is a 3-carbon VFA, makes up about 15 to 20 to 25% of the total VFA in the rumen, and is a primary source of glucose. Glucose is needed for a number of functions in the cow, including the production of milk sugar or lactose. The third major VFA is butyrate or butyric acid. This is a 4-carbon volatile fatty acid, makes up 15% of the total VFA, and is another energy source very similar to acetate or acetic acid. These are the three primary VFAs. There are several other smaller VFAs which we will not discuss today. Generally, the ratio of rumen acetate to propionate will vary, typically from 2.2 to over 3. Now, why do these VFA changes? Basically, the carbohydrates will change the VFA proportion and pH in the rumen. For example, a high forage diet, forages being such things as hay, corn silage, pasture, will have a high level of acetic acid because the bacteria that digest the forages and fiber are predominant. So we'll see 65% acetic acid and perhaps less than 15% propionic acid. However, if we shift the cow's diet to a high grain diet, we may find less than 50% acetic acid and over 25% propionic acid. We need to maintain this acetate to propionate above 2.2 as recommended by Wisconsin researchers. When it goes less than that, we may see signs of rumen acidosis. Rumen acidosis, or low pH in the rumen, usually refers to an abnormal fermentation. When the pH goes below 6, evil things can happen, as you'll see. Usually the time the cow is below a pH 6 and how low below 6 determines how severe the rumen acidosis is. When a cow experiences rumen acidosis, the amount of VFA production goes down and the amount of microbial yield also declines, which means the cow has less fuel and less protein for her utilization. Also, because of the change of pH in the rumen, it can go to the bloodstream and affect blood flow to the feet, which can lead to laminitis or animals that have sore feet. Farmers would also note a decrease in milk yield and a decrease in milk fat. So we have a change both in the amount of milk and the components themselves. The cow's health is also compromised, which may require veterinary service or affect her immune system. Another term you'll hear, championed by the Wisconsin veterinarians, is something called SARA, which stands for subacute rumen acidosis, and is felt to be the major metabolic disorder reported and seen at the University of Wisconsin hospitals for dairy cattle. Let's now leave carbohydrates and go to protein. What happens to protein in the rumen? Basically, three different fractions appear in protein. Soluble protein, also known as SP, rumen degradable protein, and rumen undegradable protein. If we understand what happens in the rumen, these terms make sense. If the protein is broken down very quickly by the bacteria, when it reaches the rumen, we call that soluble protein. It will go 200 to 300% per hour. It goes very, very quickly. It's a very fast rate and can be beneficial to jumpstart the rumen. However, if it is too fast and too excessive, it ends up going into the bloodstream and has to be excreted as blood urea nitrogen. About 30% of the total protein should be soluble protein to have an adequate amount in the rumen. The other phase would be the rumen degradable protein. This makes up about 65% of the total protein. It also breaks down to ammonia and in some cases to amino acids, which are used by the bacteria. Its rate is much slower relative to soluble protein. If the feed does not break down in the rumen, we call that rumen undegradable protein. About 35% of the feed should be targeted to be rumen undegradable protein because then the cow will digest that just like a hog or a human would digest that protein. This feed is not degraded in the rumen and goes through pretty much intact. Therefore, the amino acid profile of the rumen undegradable protein does not change. You will notice that the amount of rumen degradable and undegradable always has to add up to 100. Soluble makes up about half of the rumen degradable fraction. Fat changes in the rumen are minor compared to carbohydrates and proteins. The triglycerides found as a nutrient are hydrolyzed, which means they are broken down to fatty acids and glycerol. These fatty acids are then hydrogenated if they are in the rumen long enough to add hydrogen ions to these fatty acids to make them more saturated and have less effect on fiber digestion. Vitamin changes are also minor in the rumen. The good news, many B vitamins are synthesized by the rumen microbes. B vitamins such as niacin, biotin, thiamine, and others are made in large amounts by the bacteria, which then can be used not only by the bacteria, but also as the main source of B vitamins for the host animal. However, under some conditions, we will add extra B vitamins due to requirement or other metabolic disorders, especially niacin and biotin. Fat-soluble proteins are really not affected in the rumen. Mineral changes also are very minor. Minerals do not change in the rumen appreciably. They are not absorbed in the rumen, therefore they go down to the lower tract. Some minerals will bind to organic matter, which may affect absorption in the lower tract. Many of the organic minerals, such as zinc methionine, are rumen stable and do not degrade and go down to the lower tract. Finally, we'll talk about intestinal digestion. Some people call that the lower tract. Let's go through each of the nutrients quickly and decide what happens in the intestine. Protein basically is broken down to amino acids. Remember, the two sources of these proteins are going to be microbial protein, about 65% of the total, and about 35% from the rumen undegradable proteins are broken down to amino acids and are absorbed across the intestinal lining. By and large, the small amount of starch that reaches the small intestine can be broken down to glucose by an enzyme and absorbed as a glucose, much like in a hog or a human. Fats are broken down to fatty acids and then are absorbed as micelles and transported then as triglycerides. Remember, we said if there's too much triglyceride, the animal will store it as fat. Minerals are absorbed in the small intestine either as organically bound or tied to some other source, and vitamins are also absorbed at this point. So you can see the real action item for absorption occurs in the small intestine, very important structure in the digestive tract. Well, this concludes our description of the digestive physiology of ruminant animals. Thanks, and have a good day.